to measure a population allele and genotype frequencies tells us about the gene pool, tells us about the amount of genetic variation that's in that population. And so it's a numeric value, and so we can calculate it from data, right? So working from data to a value. So that, again, we want to be able to compare populations or to compare between time periods or different things like that. So these are the equations, if you will, for allele and genotype frequencies. So we have a fraction, so the number of copies of a specific allele versus the total number of alleles. So for example, if you have 50 of the A allele, and you have 100 total alleles, then you would have, 50 out of 100, which is equal to 0.5. So you would have a frequency of 0.5 in that population. The genotype frequency is, instead of the number of alleles, it's the number of individuals with a particular genotype. So then you want to know, for example, how many individuals have this genotype out of the total size of the population. So maybe you have 25 individuals that are heterozygous, and you have 50 total individuals. 25 out of 50 also equals 0.5. So the genotype frequency for heterozygotes would be 0.5. The allele frequency for this calculation was 0.5 for the big A alleles. So raw data doesn't necessarily come that way, in the sense that you have to work from the population. So you'll see what I mean. So if we look at an example like this, that's maybe describing a population, then we can do some calculations that way. So we have 100 plants. 49 of them are red flowered, 42 of them are pink flowered, and 9 of them are white flowered. This is in 4 o'clock plants. This is an example of incomplete dominance, which you may remember from Mendelian genetics. So you have an intermediate phenotype for the heterozygotes. So that's kind of convenient for us because we'll be able to know the genotypes based on appearance. You don't always know that because a lot of times the heterozygote looks the same. But this is a nice example where we can use that. So the allele frequency of CW, that's the white allele, I'm going to take by looking at my different individuals. So for every one of the white flowered plants, right? So there's 9 white flowered plants, but each white flowered plant has 2 of the CW alleles. So I'm going to multiply that by 2. So that represents the homozygote. And then the heterozygote, there's 42 of them, but each one of them only has a white allele. So I'm just going to multiply by 1. You don't actually have to do that, but just to see where I'm getting that value from. There's only 1 of them. And then the red flowered plants, there is 49 of them, but they have 0 white alleles. So I'm multiplying that by 0. Again, you don't have to do that, but I'm just trying to illustrate where I'm getting these values from. So that would be 0. So 9 times 2 is 18, plus 42, plus 0. That's my top value for the equation. So these are the number of CW alleles in this population. And if I have 100 total plants, each one of those plants has 2 possible alleles. So the total number of alleles is 100 times 2, which is 200. Okay. So that means that I have, let's add this together. 42 plus 18 is 50, 60 over 200. And then if I reduce that, 30 over 100. That's equal to 0.3. So the allele frequency of CW is 0.3. So I'm going to come over here and do the allele frequency of CR too, just so you can see another time. So I have 49 red flowered. So there's 49, and each of them contribute 2. so I'm going to multiply by 2. The heterozygotes have only one CR, so 42 times 1. And then the white-flowered ones have 0 of them, so I'll just put a 0 there. And then my total possible is 200 possible alleles. That's because there's 100 plants, and they each have 2 alleles. And so if I do that math, 49 times 2 is what, 98? And 42, so that adds up to 100, 140. Divide that by 200. Oops, sorry about that. And that is equal to 0.7. So my allele frequency for CW was 0.3. My allele frequency for CR is 0.7, calculated using that equation. So the R and W alleles are the only alleles that are in this population. So notice that 0.7 plus 0.3 is equal to 1.0. So the maximum possible is 1.0 or 100%. So remember I mentioned this is kind of like percentages. 1.0 is equal to 100%. So the other way that you can figure out the other allele frequency once you know one of them is to take one and subtract the other. So if I had calculated CW, I could have taken 1 and subtracted 0.3, and that would have told me what CR was. So there's CW and there's CR. So that's going to work for allele frequencies. Okay, so let's take a look at genotype frequency. So a little simpler here because the genotype represents individuals. So CW, the only individuals that have that genotype are the white-flowered. So there's 9 of them. And then the total number of genotypes is the same as the number of individuals. So that's out of 100. So this would be equal to a frequency of 0.09. If I do CRCW, so the heterozygotes, again, there's 42 of them because it lines up exactly with the number of individuals. And there's out of 100 because there's 100 individuals, so that's 0.42. And then last one, the homozygote, there's 49 of them out of 100 total. So that's equal to 0.49. Okay. And you see if you did the math, all 3 of these values would also add up to 1.0 because that's the total population. Okay, so this number plus this number plus this number would add up to 1.0. So a last question here, what is the allele frequency of a gene that is monomorphic? Well, remember we also call this if the gene is fixed or an allele is fixed. In that case, the allele frequency is 1.0. So again, everybody has that allele. So that's how you calculate allele frequencies from raw data.